In the previous session, we discussed on classification of epithelium based on the shape, the number of layers, and on the combination of shape and number of layers together. In this session, we will discuss on epithelial junctions. In the tissues where cells are loosely packed, the cell membranes of the adjoining cells are separated over most of their extent by a narrow space. Now, you can see this is the narrow space, okay, between the two cells. So this contact is sufficient to bind cells loosely together and also allow some degree of movement of individual cells. In some regions, cell membranes of adjoining cells come into more intimate contact, okay. The two cell membranes, they come in more intimate contact through specialized intercellular junctions called epithelial junctions, which serve different functions and are recognized only by electron microscope. Epithelial cells adhere strongly to neighboring cells and basal laminae subject to friction or other mechanical forces. The different types of epithelial junctions include the tight or occluding junctions, or it is also called as zonula occludens. Okay. Gap junctions are the channels for communication between adjacent cells. Adherent or anchoring junctions, that is the third type of epithelial cell junctions, are sites of string cell adhesion. They bind cells together mechanically to the neighboring cells and are also tightly attached to basement membrane by the specialized junctions. They are of three types: zonula adherens, desmosomes, and hemidesmosome. There are several types of cell adhesion molecules which are nothing but membrane proteins responsible for these specialized junctions. These proteins consist of three domains: intracellular binding protein, which interact with cytoskeleton and acts as an anchor within the cell; transmembrane protein, which protrudes through the plasma membrane; and extracellular link protein, which binds with the other cell adhesion molecules of same type, which is called homophilic binding, or with different types of cell adhesion molecules or extracellular matrix, which is called as heterophilic binding. Cell adhesion molecules are classified into two groups: calcium dependent and calcium independent. Calcium dependent cell adhesion molecules include adherens, which cause adhesion via homophilic binding. Three important types of adherens are seen: E adherens in epithelia, P adherens in placenta, and N adherens in nervous tissue. Integrins are involved in binding of cells with components of extracellular matrix such as fibronectin. Selectins, they are present in leukocytes. They bind to carbohydrates present on other cell surfaces. However, the binding is relatively weak. The calcium independent cell adhesion molecules include neural cell adhesion molecules, which are expressed by nerve cells, and intercellular cell adhesion molecules, which are expressed by leukocytes. Let us now see the details of each epithelial junction. Okay, zonula occludens, also called as occluding junction or tight junction, is present just below the apical surface of the epithelial cell. It is formed by a network of sealing strands extending along the entire circumference of the cell. These strands are composed of transmembrane proteins present in the plasma membrane. The outer layers of plasma membrane of the adjacent cells join with each other and obliterate the intercellular space between them. If you can recall, the transmembrane proteins were discussed in the cell membrane. Okay, so if you don't recall or remember, please go back and watch the lecture once again. Okay, coming to the functions, The occluding junctions, okay, binds the adjacent cells with each other. It acts as a barrier as it prevents the movement of substances from the lumen of the viscera into the intercellular space. Certain substances enter the cell by diffusion or active transport and pass through the tissue. Thus, cells can control the movements of substances across the tissue. The apical surfaces of the cells have proteins like receptor protein performing specialized functions. These are absent on other surfaces of the cell. Okay, so the receptor proteins are present only on the apical surfaces and they are absent on the other surfaces. So what is happening here? These occluding junctions, okay, they prevent these receptor proteins from entering into the other surfaces. Okay. The second type of the epithelial junction, that is the gap junction, also called as communicating junction, is important in cell-to-cell contacts that facilitate communication between cells via electrical or metabolic coupling. They connect the plasma membrane of adjacent cells. They are made of hexagonal intercellular channels called the connexons. The alignment of two connexons from the adjacent cells, okay, across the gap between the two plasma membranes results in the complete intercellular gap junction channel. Each connexon is formed by 6 copies of transmembrane proteins called connexins. The gap junctions are less in epithelium and more in cardiac and smooth muscles. They have an important role in the functioning of cardiac and smooth muscles, where the signals pass through these junctions from one cell to another. This results in the simultaneous involvement of large numbers of contractile units during excitation. Gap junctions allow the passage of ions and small molecules. Coming to the anchoring or adhesive junctions, which is the third type of epithelial junctions, they are most abundant in tissues that are subject to constant mechanical stress, such as epidermis of the skin. They attach the cells and their cytoskeletons mechanically to their neighboring cells or to the extracellular matrix. They are of 3 types: zonula adherens, desmosome and hemidesmosome. Zonula adherens and desmosomes, okay, these two, they bind the 2 cells, okay, adjacent 2 cells, but hemidesmosome, it binds the cell to the basement membrane or the extracellular matrix. Functions of the anchoring junctions: they bind the neighboring cells. These attachments are stronger than the occluding junctions. It allows a group of cells to function as a cohesive unit. Going in detail, the 3 adhesive junctions, okay, to start with zonula adherens or adhesive belt, they are present as a band below the zonula occludens and completely surround the circumference of cell. Okay, so let us look at the structural arrangement. When we take, okay, we will look into only this part of the junction, okay, cell membranes at where the zonula adherens is present. Okay, there are, okay, you can see 2 adjacent cell membranes and the intercellular space between the 2 membranes. On either side is the cytoplasm inside the cell 1 and cell 2. Okay. Now the transmembrane protein of adjacent cells bind together to form adhesive junction outside the cells. Okay. So they bind outside the cell. Two transmembrane proteins bind together in the intercellular space, and inside the cell, okay, inside the cell, it gets attached to the actin filaments. The actin fibers are linked to transmembrane protein via actin binding protein. The second type of anchoring junction, the desmosome or macula adherens, are the most common type. These are present where strong anchorage is needed. It is a small circumscribed area of attachment. Okay. To look at the structure, okay, here again, if we take this part, okay, so we will see, okay, the two cell membranes, okay, the intercellular space between the two membranes, and on either side the cytoplasm inside the cell 1 and cell 2, okay. The transmembrane protein, okay, is formed by desmoglein and desmocholine, which are two types of adherens, okay. The extracellular part of the transmembrane protein, that is this part in the intercellular space, okay, gets attached with the similar transmembrane protein of the adjacent cell. The intracellular part of the transmembrane protein binds to the cytoplasmic plaque made of protein desmoplakin. This cytoplasmic plaque is attached to the intermediate filaments. Desmosomes provide resistance against the shearing force between neighboring epithelial cells. Hence, they are more abundant in the epithelium, subjected to mechanical stresses such as epidermis of the skin. Hemidesmosome, the third type of anchoring junctions, binds the basal surface of the cells with basement membrane. They resemble half of a desmosome. In contrast to desmosomes, the transmembrane proteins in hemidesmosomes are integrins, okay, instead of adherens. Looking into the structure, there is cytoplasm inside the cell, the basal surface of the cell membrane, okay, that is, we are looking at this part, okay, here at the base of the cell. So there is the cytoplasm on the inside of the cell, then the base of the cell, okay, the cell membrane at the base of the cell. So here the integrin, okay, the integrin outside the cell binds to the basement membrane, and inside the cell it binds to the intermediate filaments. Epithelial tissues are relatively labile structures whose cells are renewed continuously by mitotic activity and stem cell populations. The rate of renewal varies widely. Okay, it can be fast in tissues such as intestinal epithelium, which is replaced every week, or slow as in the large glands. In stratified epithelial tissues, okay, the stem cells are present in the basal layer and mitosis occur only within the basal layer in contact with the basal lamina. In some functionally complex epithelia, stem cells are located only in restricted places, like at some distance, okay, from the transit amplifying cells and differentiating cells. Okay. For example, if we take the epithelium lining the small intestine, it is derived completely from stem cells found in the simple glands which is present between the intestinal villi. Okay. In the epidermis of thin skin, stem cells are located along the wall of hair follicles. Epithelia are normally capable of rapid repair and replacement of apoptotic or damaged cells. In some larger glands, most notably the liver, mitotic activity is normally rare but is actively renewed following major damage to the organ. When a portion of liver tissue is removed surgically or lost by the acute effects of toxic substances, okay, cells of the undamaged regions quickly begin active proliferation and a mass of liver tissue with normal function is regenerated. Now, to conclude, in this session, we discussed different types of epithelial junctions like tight junctions, gap junctions and adhering junctions, and we also discussed on renewal of epithelial cells. So in the next session, okay, we will be discussing on clinical anatomy of the epithelial tissue. Okay, so see you there. Before you leave, share your lessons learned down below without fail. Welcome back.